welcome to Bioholic Friday class with your educator Olivia. After discussing the auxin, gibberellin, cytokinin and ethylene, it's the time to learn about the abscisic acid. So today's class is totally based on abscisic acid plant hormone. Okay? As this is the fifth important plant hormone, this class is the last class of the chapter plant hormones. Watch the video attentively because there is again a self-assessment test on today's class. So let's start. The abscisic acid is often referred to as an inhibitory rather than stimulatory hormone, and also it is called as the plant stress hormone. And by its name it can be understood clearly that it is slightly acidic in nature. And the chemical formula of ABA is C15H20O4. Now why it is called as inhibitory hormone? Because abscisic acid functions as a plant growth inhibitor, including seed and bud dormancy, the control of organ size and stomatal closure. And abscisic acid is also known as dormin as it induces dormancy in buds, underground stems and seeds. Okay? And why it is called as stress hormone? Because the synthesis of the hormone is stimulated by environmental stresses like drought, waterlogging, etc. and it plays an important role in tolerating abiotic stress. But how? Actually, abscisic acid stimulates closure of stomata during high salinity water stress and reduces water loss by transpiration. ABA interacts with other phytohormones like jasmonates, nitric oxide and signaling molecules to induce stomatal closure. And ABA also induces seed dormancy and thereby helps seeds to withstand desiccation and other unfavorable conditions for the growth. And because of this, seeds can be kept in storage for a longer duration. ABA plays an important role in the growth and modification of root during nitrogen deficiency and drought, and it regulates the gene expression required for root growth maintenance and water uptake. Moreover, ABA plays essential roles in signal transduction pathway during the stress responses, and ABA stimulates long-term growth by regulating stress-responsive genes. That's why, because all of these reasons, ABA is known as the plant stress hormone. But now I want to ask you something. Which hormones are called as stress hormones in case of human? If you know the answer, write in the comment section. But if you don't know the answer, then go and check the playlist of chemical coordination and integration. There you will get your answer definitely. And also don't forget to check that why those hormones are called as stress hormones in humans. Now moving on the next slide. The history about abscisic acid invention. In 1963, a substance that promotes abscission in cotton fruit was invented and called abscission 2. On the other hand, another substance is found that promotes bud dormancy and that was called as dormin. In 1964, it became evident that both the abscission 2 and dormin are same group of hormones. And later the name was changed to abscisic acid. Now the biosynthesis of abscisic acid. Abscisic acid is an isoprenoid plant hormone which is synthesized in the 2-C-methyl D-erythritol 4-phosphate MAP pathway. Zeaxanthin is the first committed ABA precursor. A series of enzyme-catalyzed epoxidations and isomerizations via violazanthin and final cleavage of the C40 carotenoid by a dioxygenation reaction aids the proximal ABA precursor xanthoxin. Xanthoxin further oxidized to ABA via the abscisic aldehyde. In the figure below, the reactions are depicted here. So from the xanthoxin, the abscisic aldehyde is formed by the action of xanthoxin dehydrogenase, and in the final step, the abscisic aldehyde is then converted into abscisic acid by the action of abscisic aldehyde oxygenase. Now the locations. Abscisic acid released during desiccation of the vegetative tissues and when roots encounter soil compaction. They are synthesized in mainly green fruits at the beginning of the winter period, and they are also synthesized in maturing seeds establishing dormancy. They are mobile within the leaf and can be rapidly translocated from the roots of the leaves by the transpiration stream in the xylem. They are produced in response to environmental stress, as I said, such as heat stress, water stress, salt stress, and they are synthesized in all plant parts, for example, the roots, flowers, leaves, and stems. ABA is synthesized in almost all cells that contain chloroplasts and amyloplasts. Now, how the ABA action is mediated? Actually, in the absence of ABA, the phosphates ABI1 insensitive 1 inhibits the action of SNF1 related protein kinases. The ABA is perceived by the pyrobacter resistance 1 or PYR1 and PR1 like membrane proteins. So in presence of ABA, on ABA binding, PYR1 binds to and inhibits the ABI1. So then the SNRK2s are released from inhibition and they activate several transcription factors from the ABA responsive element binding factor or ABFs. These ABFs then go on to cause changes in the expression of a large number of genes. Around 10% of plant genes are thought to be regulated by ABA. So thus, the mechanism of action of ABA is mediated. Watch this figure to clear your concept. Here, as I said that in absence of ABA, the PYR1 is inactive, therefore it cannot inhibit the ABI1. And thus ABI1 is inhibiting SNRK2s. But in presence of ABA, the PYR1 binds to ABI1, thus inactivate its inhibition on SNRK2s. Now, the SNRK2s activates the ABFs, thus changes in transcription occurs. Okay? Now the physiological functions of ABA. First of all, the bud dormancy. Abscisic acid induces dormancy of buds towards the approach of winter. Next, the seed dormancy. Seed dormancy is mainly caused by the abscisic acid. Dormancy allows seeds to tolerate desiccation and extremes of temperature better. The buds as well as seeds sprout only when abscisic acid is overcome by gibberellins. So gibberellins and abscisic acid have opposite effects. Thirdly, the stoppage of cambium activity. Formation of abscisic acid stops mitosis in vascular cambium towards the approach of winter. And the abscission. Abscisic acid promotes abscission of flowers and fruits, and because it promotes abscission, thus it is named as the abscisic acid. Now the leaf senescence. Its excessive presence stops protein and RNA synthesis in the leaves and hence stimulates their senescence, though the leaf fall is actually promoted by the ethylene. Sixth is the transpiration. During desiccation and other stresses, abscisic acid is rapidly synthesized. The inhibitor causes closure of stomata and hence prevents transpiration. Seventhly, the resistance. Abscisic acid increases resistance of plants to cold and other types of stresses. It is therefore known as the stress hormone. And the starch hydrolysis. Abscisic acid inhibits gibberellin-mediated amylase formation during germination of cereal grains. In small quantities, abscisic acid is known to promote flowering in some short-day plants, for example strawberry, blackcurrant, etc. And ABA has been found to induce parthenocarpic development in rose also. Rooting of stem cutting is promoted in some cases by abscisic acid. For example, the beans, AV, poinsettia, and ABA is important for controlling of growth. It is antagonist to gibberellins and counteracts the effects of other growth promoting hormones like auxins and cytokines. Therefore, it keeps their activity under check. By controlling growth, ABA plays an important role in seed development and seed maturation. Normally, it inhibits seed germination, growth of excised embryos, growth of duckweed and other plants. And lastly, the practical applications of abscisic acids. Application of minute quantity of abscisic acid to lips shall reduce transpiration to a great extent through partial closure of stomata. It conserves water and reduces the requirement of irrigation. Photosynthesis is reduced to a lesser extent. And abscisic acid is useful in introducing flowering in some short day plants kept under unfavorable photoperiods. Use of abscisic acid promotes rooting in many stem cuttings. And lastly, but not the least, abscisic acid can be used in prolonging dormancy of buds, storage organs, and seeds. Now, the fun fact about abscisic acid is that fungi make use of abscisic acid too. Yes, some plant pathogenic fungi also produce abscisic acid. This helps the fungus by suppressing plant defenses. So that is the end of the chapter. But before ending the video, I want to announce that on the 17th December 2020, that is the next Thursday, I will take a mock test on this chapter, plant hormones. So start your preparation, and if you missed the previous videos on plant hormones, then go and check the playlist of plant hormones. I will also provide the link in the description box. Okay, so please check, and the mock test will be on 25 marks. So best of luck, and on that day I will also discuss the NEET questions of last 20 years, that is from the year 2000 to 2020, based on this chapter, and I will also provide you the solutions. So mark the date of the Bioholic Extra class on 17th December 2020, that is the next Thursday. And from the next Friday, I will start another chapter, that is the photosynthesis. So if you have any queries or doubts, you can ask me through our mail also. And also don't forget to check the self-assessment test link and ACQ link in the description box. And if you are new here, then subscribe our channel and attend our regular classes. Don't miss the full biology package, absolutely free. So see you in the next Thursday. Thank you. Keep watching Bioholic.